All right, let's begin today's session. Okay, so I am Surasree. I will be taking this live session of week 2 of the NPTEL course Experimental Biochemistry. So let's begin. So in this session, uh, the topics that I'm going to cover is amino acids and their properties, the determination of isoelectric point of the amino acids, and titration of the amino acids. Okay, so let's begin our discussion with amino acids. Amino acids, these are the building block of protein molecules, right? So amino acids, they have this carboxyl group and this amino group, both joined to the alpha carbon. So not only these amino acids have the amino group as well as the carboxyl group, but they also contain one side chain which is denoted by this R. So this is the side chain group. Now these amino acids, they are joined together by peptide bond to form proteins. So they form peptide bond. And these amino acids are linked by peptide bond. Many amino acids linked together by peptide bond, they form this protein structure. Now this R group, this side chain, can vary between different amino acids, and according to this variability of this or the presence of different side chain groups, we have 20 common amino acids. So we have 20 common amino acids which have different R groups. Right? And amino acids are usually denoted by a 3-letter code or 1-letter code. So these are abbreviations by which we denote amino acids. So uh, we have, for example, in our body amino acids like alanine, valine, isoleucine, tryptophan. So all these amino acids, we have assigned different codes to them, and these are either 3-letter codes or 1-letter codes. All right, so this is the general structure of amino acids. Now let's look at how the 20 common amino acids look like, and in the meanwhile we will also discuss the different properties of the amino acids. For example, like what is the size and shape of the amino acids, what is the charge of the amino acids, what is the polarity, hydrophobicity. These different characters we will also discuss while we are discussing the 20 common amino acids. So first let's talk about these aliphatic amino acids. So you can see that aliphatic amino acids, the first of all, the simplest amino acid is the glycine. The simplest amino acid is the glycine. And you can see the structure. The structure is like you have this carboxyl group, there is this amino group, but there is, what is the R group here? The R group is only a hydrogen, right? So here in this carbon there are only 2 hydrogen attached to them, right? So here that is why this glycine is actually considered the simplest amino acid, because as this side chain it has only a hydrogen. Now next, coming to alanine. So for alanine, here we can see that the side chain molecule is a methyl group. So here the R is a methyl group. Now next, coming to valine. So these are all aliphatic amino acids because they have aliphatic groups as their side chains, right? So let's come to valine. Valine has this kind of a structure. This is the R group, right? This is the R group of valine. Next, coming to leucine. Leucines, and this is isoleucine, and this one is a proline. So now you can see that these amino acids, they have their full names, but we are denoting them in their abbreviations, which are either a 3-letter code or a 1-letter code. So for example, glycine can be denoted by G, alanine can be denoted by ALA or A. So similar to that. And proline, let's talk about proline here. Proline is called an amino acid because if you look at the structure, you will see that the side chain is actually linked by this ring structure with the alpha carbon, right? So this one is this one is the alpha carbon, right? And the side chain and the amino group. So this is actually not an amino group, but rather it's an amino group. So which is why this proline is called an amino acid. And also remember, because of its different structure, proline gives rigidity when it is present in proteins. So when it is present in a particular structure in a protein, then what happens, this proline amino acid gives rigidity to that structure, right? So these are the aliphatic amino acids: glycine, alanine, valine, leucine, isoleucine, proline. These are considered aliphatic because they have aliphatic side chains. So there will be some other amino acids as well which will contain aliphatic side chains. We will see. So next are these sulfur containing amino acids, right? So the amino acids are methionine and cysteine. Methionine is this mate, and cysteine is, which can be also denoted as cis. And the structure that is like below this particular structure, this is actually a cysteine molecule. So what is a cysteine? So it's not an amino acid, rather it's a peptide formed by 2 amino acids, which are 2 cysteine molecules. So 2 cysteine molecules, I'm sorry, I'm mistaken. It's not a peptide. Please don't confuse. It's not a peptide, but when these 2 cysteine molecules are joined by disulfide bridge or disulfide linkage, then it forms a cysteine. Okay, this kind of a structure is very commonly observed in proteins, and the importance of this kind of a disulfide linkage, it actually creates various shapes and various conformations in proteins. Okay, so it is important for that reason. And so this is something you can see, that 2 cysteine molecules. So this is one molecule. If you look at this structure, this is one molecule and this is another cysteine molecule. So 2 cysteine molecules are joined together by the disulfide linkage, which is this one. So that forms a cysteine. And please don't confuse, it is not a peptide structure. It's not a peptide bond. but rather it's a disulfide bond. And this is also one important amino acid, methionine. And you might know that all proteins, when they are translated in the ribosome, they begin with this particular amino acid, or they start with this particular amino acid called methionine, right? So, and these amino acids, these you can also link to aliphatic amino acids, because these also contain aliphatic side chains, but they are also distinct in this way that they contain sulfur group in them, okay? And which is why proteins are known to have sulfur in them. So these are the amino acids which provide sulfur in the proteins. Okay, next moving on to aromatic amino acids. Okay, please let me know if you have any doubts. Please, you can interrupt anytime. Okay, you can ask me in the chat, or you can unmute your mic and ask if you have any doubt. All right, next coming to aromatic amino acids. So these amino acids are different from the aliphatic ones because they have aromatic side chains, and these are phenylalanine, tyrosine and tryptophan. Okay, you can look at the structure of these amino acids. The phenylalanine contains this phenyl group linked to a methyl group. So here there is a methyl group, and here the phenyl ring is present. And next is tyrosine. So tyrosine has a phenol group, right? Tyrosine has a phenol as its side chain. And tryptophan, this is a ring which is called indole ring. So tryptophan contains this indole ring. This is phenol group, and this one is phenylalanine. Right? So these are the aromatic amino acids, and remember, when we are checking the, for example, when we are doing spectrophotometric analysis, so when we are checking absorbance of a protein, then these amino acids are the key players, because these amino acids gives the absorbance at 280 nanometer. So absorbance at 280 nanometer. So when we do spectrophotometric analysis of proteins, or when we try to measure the concentration of proteins using a spectrophotometer, so we check the absorbance of proteins at 280 nanometer wavelength, and these are the amino acids which are responsible for giving this absorbance at 280 nanometer. And remember that if the protein does not have this kind of aromatic amino acids, if the protein is completely devoid of aromatic amino acids, then it will not show any such absorbance at 280 nanometer. Alright, so these are the aromatic amino acids. Now next, coming to acidic amino acids, or the amino acids which are also negatively charged. So in physiological pH. Okay, so I'm writing it here, negatively charged amino acids. And these are called acidic amino acids because they have an extra carboxyl group. So this one is the alpha carboxyl group, right? The one which is present in all amino acids. But this particular one here, this is the R group. So this carboxyl group is extra in this amino acid, so which is why it is an acidic amino acid, or it can stay in a negatively charged form in the physiological pH. So this one is aspartic acid, written as asp or D, and this one is glutamic acid. Here also this one is the R group, and Not only that, like there are here there is a methyl group associated with this carboxyl group, and there are two methyl groups associated with this carboxyl group. So that is the difference between aspartate and glutamate, or aspartic acid and glutamic acid, right? So they have these extra carboxylic acid group, and these are the acidic amino acids, and there are also their amines. So if we uh, replace this carboxyl group with the NH2 group, right? So these are the amines that we get, which is asparagine and glutamine. So look at these structures, aspartate and asparagine, they are very similar. Just in place of this hydroxyl group, you have this amino group, right? And here in place of this hydroxyl group, you have this particular amino group in glutamine, glutamate and glutamine, right? So these are the amines of these acidic amino acids. Any doubt so far? All right. Okay, so moving on. These now coming to the basic amino acids. So basic amino acids are the ones which are positively charged in physiological pH. So always remember, when you're talking about positively charged, negatively charged, always remember you have a reference pH. So in which pH it is positively charged or in which pH it is negatively charged. Because as we will see later on that uh, with the change in pH, the charge of the amino acid will change, and which we will study in titration. So which is why it is important that basic amino acids are positively charged in physiological pH. Now because they are basic amino acids, so they have an extra amino group with them, right, which is present in the side chain. So just like the acidic amino acids had an extra carboxyl group with them, so here the basic amino acids have an extra amino group with them. And so for example, like this, you can see this is the R group of arginine, right? This is the R group of arginine, and here you can see that there are multiple nitrogen or amino groups present, so that makes the amino acid basic. Arginine, next is lysine. Lysine also you can see similar structure, and this is histidine. In histidine, the ring structure is so there is a ring structure which also contain extra amino groups, right? So these are the basic amino acids: arginine, lysine and histidine. And these amino acids, the hydroxyl group containing amino acids, they are serine and threonine. So you can see that there are hydroxyl groups present in these amino acids, serine and threonine. So that's all for the 20 common amino acids we have. And you have looked at the how what is their structure, how they look like, and you have to remember these structures, you have to know these structures quite well, because when we will be studying about proteins, or when we will be studying about the titration of amino acids, we have to remember what is their structure, what is the R group that is present in these amino acids, and how these amino acids behave. Now, talking about behavior of these amino acids are amphoteric. So what do I mean by amphoteric? Amphoteric means these amino acids have both positive and negatively charged groups in them, right? So if we look at this central structure, look at this particular structure here. This structure here, you can see that this is a general structure of an amino acid, right? And here you can see that the carboxyl group is in CO minus form, right? Carboxyl, so it's a carboxylate ion, and here the NH2 group or the amino group is present as NH3 plus, right? So this NH3 plus and the CO minus, these two charges balance each other. So in this particular state of this amino acid, there is no net charge on it, right? Because the NH3 plus and CO minus, they balance each other, right? So which is why this particular structure is called a Zwitter ion. It is a Zwitter ion because there are two charges. One is a positive charge, another is a negative charge, and then also there is no net charge on the molecule because they balance each other or they cancel each other, right? So this is a Zwitter ionic form of the amino acids. And remember that all the amino acids, they can exist in this Zwitter ionic form in a particular pH. So the pH in which the amino acids exist in this Zwitter ionic form, that pH is called the isoelectric point. That is called the isoelectric point because at this particular pH, the molecule carries no net electric charge, and also this molecule does not move in any electric field because it does not contain any net charge, right? So it does not move in an electric field, and this point, that is why it is called the isoelectric point, right? So in this point, the amino acid exists in a Zwitter ionic form. Now this point, the point that is the pH in which the amino acids exist in Zwitter ionic form, differs from amino acid to acid. Why? Because these amino acids, as you have seen just now, that they have different structures, right? So they will not ionize or they will not form a Zwitter ion at any particular pH, at any single pH. So they all have different isoelectric points based on which they form Zwitter ions at different pH, right? And we will also see that not only this Zwitter ionic form, but amino acids also exist in these two forms, as a cation or as an anion, right? So how does these structures form? So for example, if you have, or if you begin with this cationic form, which exists in low pH conditions, right? So where what you have is, first of all, in low pH condition, you know that there are the hydrogen ion concentration is very large in this particular solution because it has low pH, right? So in this situation, all the groups on the amino acid will be protonated, right? So you can see that this amino group is protonated. It is existing in an NH3 plus condition, and the carboxyl group is also protonated. So it is in this COH condition, right? So here all these, so since it is existing in a low pH condition, so all the groups are protonated, and the amino acid exists as a cation, right? So what is the charge here? The charge is +1. What is the net charge on this amino acid? The charge is +1, right? But however, when you increase the pH, right? So for example, if you are adding NaOH, or if you are adding any alkali or base in the solution, then what happens? It slowly increases the pH, right? So as you increase the pH, if you go in this particular direction, right, then what happens? At a particular pH condition, the first group that will lose the proton will be this carboxyl group, the alpha carboxyl group, okay? The first proton will be lost from the alpha carboxyl group as we increase the pH, right? So it will come to this situation where it will have COO minus. So here the NH3 plus group at this pH condition will not lose the proton because it has a higher pKa value, which is the dissociation constant, right? So at this situation only the carboxyl group will release the proton because it has a less pKa, and the net charge on this molecule will be 0. So this is the net charge we are talking about. The net charge on this molecule will be 0. Now as we again increase the pH, next what will happen, this alpha amino group will also lose its proton, or it will be deprotonated, right? So here this alpha amino group now comes as now exists as an NH2 group, right? Not as an NH3 plus. And since this NH3 plus group has a higher pKa value, so which is why it is dissociating the proton, or it is releasing the proton at a higher pH value. And at this condition the amino acid exists as an anion, right? So here then what is the net charge? -1, right? The -1 charge coming from the CO minus ion, or the CO minus group, right? So here you can see that this particular amino acid is existing in 3 different ionic stages. The first one is cation, where the charge is +1, and this is at a low pH value. Next, when it loses the proton of the carboxyl group, it becomes 0 charge, so there is no net charge on this molecule, it exists as a Zwitter ion. And next what happens when you increase the pH more, then at a higher pH this amino group also loses its proton and it becomes negatively charged, so the net charge becomes -1, right? So amino acids can exist in these different forms, these different charges, right? Ionic forms. Now the next thing that we will not discuss here, but we will just revisit, that is a Henderson-Hasselbalch equation. Why we are revisiting this? Because we will need this Henderson-Hasselbalch equation to calculate the isoelectric point of amino acids, right? So this is something that we had discussed in the previous session, that was in the last week. And if anybody wants the video of the previous session, you can find it in the course website under the problem solving session tab. The YouTube link of this video is already provided, and in the at the end of today's slide also I have included the link of the YouTube channel where I will be uploading these videos, so you can go there and find it. So Henderson-Hasselbalch equation we had discussed in the previous lecture, and this equation is simply this particular equation, which is pH equals to pKa plus log of concentration of the conjugate base divided by the concentration of the acid. And this is when a weak acid is dissociating. So this Ka is the dissociation constant, and when we put log on both sides we get this particular equation. That is the Henderson-Hasselbalch equation. Now I need you to remember this so that we can calculate the Pi of particular amino acids. Okay, so now here we will discuss the titration of the simplest amino acid, which is glycine. Now if you remember the structure of glycine, the alpha carbon, here this alpha carbon contains 2 hydrogen atoms and 1 NH3 or NH2 group and COH group, right? Here look at this first structure. Take a look at this first structure. So one thing that one side information that I'm just telling you, that is glycine is the amino acid where the alpha carbon is not a chiral carbon. Am I right? Because the alpha carbon contains there's no side chain as such. So the side chain is hydrogen atom, so which is why it's not a chiral carbon, the alpha carbon. All right. So moving on, this particular structure that you see here, the structure that I have squared, this is a positive state of the glycine, positively charged glycine. So where you have NH3 plus and COOH, right? And so the net charge is +1, which is why I have written here that it is glycine plus, right? And as I've told you that this will exist in a low pH condition. So when the pH is low, glycine will exist as a glycine plus molecule or a positively charged molecule. Now as we increase the pH, then what will happen, this carboxyl group will lose the proton first. So remember that alpha carboxylic groups, they lose their proton first because they have low pKa value, right? And if we denote that as pK1, the dissociation constant of this carboxyl group, the proton of this carboxyl group, then what we can write based on the Henderson-Hasselbalch equation. So first of all, this molecule is now a zwitterion, right? So there is no net charge, so I have written glycine 0, right? I have denoted it as glycine 0 because it has no net charge. The positive and the negative charges, they cancel each other. So if we now write the equation, Henderson-Hasselbalch equation, which you know that pH equals to pKa plus log of concentration of the conjugate base by the acid. So you will write it as Pi equals to pK1 plus log of concentration of glycine 0. So glycine 0 is here the base, right? Because this one is the acid. Why this one is the acid? Because this is releasing the proton, right? So we are considering it as the acid. So I have written it here in the denominator, glycine plus, and this one is the conjugate base. So we have written it in the numerator, glycine 0, right? So concentration of glycine 0 divided by concentration of glycine plus. That is equation 1. Now next, I told you when we are increasing the pH. So at high pH condition, what happens? This structure can now be formed as the amino group is now again releasing its proton, right? So the amino group is releasing its proton. Therefore, now the glycine is negatively charged, right? And we are denoting it as glycine negative, right? Because the net charge on it is -1, right? So now this occurs at a higher pH value because the NH2 group, it has a higher pKa value. And if we consider it as pK2, then we might write this equation, Henderson-Hasselbalch equation, as Pi equals to pK2 plus log of concentration of glycine minus. So here, this one is the base. In this equation, this one is the base because this particular form is the acid because it is losing the proton, right? And we know that acids are proton donors, right? So that is why glycine 0 we have written in the denominator and glycine negative we have written in the numerator, right? So if we can write it as Pi equals to pK2 plus log of concentration of glycine minus divided by concentration of glycine 0, and we have denoted it as equation 2. Now please let me know if you have any confusion till this part in these two equations, how these equations are formed. Okay, all right. If the other two people, if you do not also have any confusion, then we are proceeding. So what we will now do is to calculate the Pi. Now we will simply add these two equations, that is add 1 and 2, and what we get is 2pi equals to pk1 plus pk2 plus log of glycine 0 into glycine negative divided by glycine 0 into glycine positive. So we are actually adding these two values, right? Log of a plus log b is giving you log of a into b, right? We are getting this value, and here obviously we can cancel these two terms, glycine 0, right? And what we get here is that 2pi equals to pk1 plus pk2 plus log of glycine negative by glycine positive. And in this condition, when this glycine negative concentration is equal to the glycine positive concentration, then what we can say is 2pi will be equal to pk1 plus pk2 plus log of 1, right? Because if these two concentrations are same, then we will get log of 1, and log of 1 is 0, right? So therefore we will get this particular equation, which is 2pi equals to pk1 plus pk2, right? And that from that we can easily determine the pi, which will be pk1 plus pk2 divided by 2, right? So that is the equation with which we determine the isoelectric point of amino acids. Pi equals to half of pk1 plus pk2. What is pk1? pk1 is the dissociation constant of the carboxyl group proton, and pk2 is the dissociation constant of this amino group proton, right? So by this is the equation which you will need to remember for solving the sums for the titration problems, right? That pi equals to half of pk1 plus pk2. All right. So no doubt so far? All right. Okay. So if that is okay, then we will proceed with the how this titration of glycine actually look like when we do this experiment of titration of glycine. So how it will look like experimentally, right? So glycine, when we titrate, so how do we titrate? We titrate with a alkali, right? And so when we are adding an alkali, so here you can see in the x-axis we are adding the OH minus or the alkali we are adding, right? So now when alkali or base we are adding, so the at the beginning when the solution is in a very low pH condition, right? So just starting from here, so we can see at the beginning, so this y-axis is the pH value, right? pH is increasing along the y-axis. So in the beginning when there is a low pH condition, I told you that in a low pH condition the amino acid will exist as a cation, and this is the structure that is predominant, that forms in this low pH condition, right? When you have the +1 charge on the amino acid glycine. Now as we increase the pH, so as we are adding alkali to this solution, then what happens, so the carboxylic acid group, I told you that it has a pKa less than 6. So it is approximately in this range of 2 to 3, the pKa of the carboxylic acid. So of course this carboxylic acid group, it will lose the proton at an acidic pH, right? And here we can see the pK1 of the glycine is 2.34. So when it reaches the pH 2.34, you can see here this is actually 1, this is 2. So when the glycine is reaching pH 2.34, when we are adding alkali and the pH reaches around 2.34, then the glycine loses its proton from the carboxylic group, right? And now it exists as a zwitterion, of carrying no net charge, right? So sorry. So as we're increasing the uh, pH, now the glycine loses its proton from the carboxylic acid group, and again further as we increase the pH, and you can see that the pK2 of glycine is approximately 9.60. So these amino groups, they have pKa more than 8, greater than 8. So of course they will lose the proton at a basic pH, right? So you can see that in this particular pH, that is pH of 9.6, when it reaches the pH of 9.6, the glycine can lose its proton from the amino group, and now it exists in a anionic form carrying -1 net charge, right? So what happens here is that this pK1 you can determine 2.34, pK2 is 9.60. Now how can you determine the pi? So as I had mentioned, pi equals to half of pK1 plus pK2. So can you therefore it is like half of 2.34 plus 9.60, right? So which will give you approximately 5.97, which is written here, the pi of the glycine molecule. And this is the condition where the glycine will exist in this zwitterionic form, right? This is the pH where the glycine will exist in zwitterionic form. And you can see here in this particular graph that first of all, this graph is like it's going from it's increasing, and then suddenly the pH, as the pH is increasing in this particular region, you can see in this particular region you can see that the pH is increasing. So it is increasing from, it is 3, 4, 5, 6, 7, 8, 9. So from pH approximately 5, sorry, from pH approximately 5 to pH 9, you can see that even though pH is increasing, there is no such change in this curve of titration of the glycine, right? So the curve is going straight, it's going straight upwards, and there is no such change. So this is actually the buffering zone, where it is resisting the change, right? And this is called the buffering zone. And then what happens after pH moves from 9 to 10, so when the pK2 value is reached, then what happens, then this glycine again loses its proton from the amino group and it becomes negatively charged. So in this change of the glycine from +1 to 0 to -1, you can calculate the pi value, and which is coming as 5.97. So you can see that the pi value is approximately 6. So in the physiological pH, which is 7.4, glycine will exist as a zwitterion, right? Because in this 7.4 pH, you can see that there is no such change in the net charge of glycine, right? Because this forms the buffering zone, and therefore at a physiological pH, glycine will exist as a zwitterion, which is why, so if you take glycine and if you dissolve it in water, normal water, then it will also exist as a zwitterion, because normal water usually has a pH of 7. So glycine will exist as a zwitterion in this normal water also. So if we have to do titration of glycine experimentally, then what happens that first of all you need to protonate glycine, right? So you need to reach this particular state of glycine, and which cannot be reached in normal water, because if you dissolve glycine in normal water, it will exist in this zwitterionic form and not in this protonated form. So if you have to titrate glycine, so that is why what we do is, when we begin titration of glycine, we add equimolar amount of hydrochloric acid, to begin the titration, to begin this protonation, because otherwise glycine will not be protonated and the initial pH cannot be, or the pKa cannot be calculated, right? So to begin titration of glycine, we need HCl, equimolar amount of HCl is given, so that glycine is protonated at low pH, right? And then we can begin the titration of glycine. All right, is that clear? Okay. All right. So next we will discuss about the pKa of several amino acids. So these are the 20 common 20 common amino acids that are present in our body, present inside our proteins, right? And you can see that there are 3 pKa values. You can see that there are 3 columns. One is a pKa of the alpha carboxyl group, one is a pKa of the amino group, another is a pKa of the R group or the side chain, which we can also denote as pKr, right? So in the previous discussion of glycine, we were talking about only 2 pKa values, right? The pK1 and pK2, and pK1 corresponds to the pKa of the alpha carboxylic acid or the alpha carboxyl group, and the pK2 corresponds to the pKa of the amino group, right? But here we also have a pKa of the R groups, where the R groups are polar or the R groups are charged, right? So here you can see that the or there are particular the structures which can ionize, so that is why they also will have a pKa value, the particular R groups, the side chains. So which is why we have 3 pKa values for these amino acids, like for example cysteine, aspartic acid, glutamic acid, arginine, histidine, lysine, right? Because they are all amino acids which contain ionizable side chains or ionizable R groups. So we have a pKr value of them as well. And here if you look at this table, you can see that the that the pKa of this alpha carboxyl group, it usually ranges between 2 to 3, right? So approximately in the range of 2, 1.8, 2.21, 2.63. So at pH greater than 3, they will all lose the proton, right? So they retain the proton, the alpha carboxylic groups retain the proton only in low pH condition, very low pH condition, which is less than approximately 3 or something, right? Because their pKa is approximately 2 to 3. So after pH 3, if you increase the pH, they will lose the proton, right? And that is why they lose the proton first during titration. Next is the pKa of the amino groups, and you can see here the pKa exists here, which is like 9.6, 9.6, 10.6, 9, 9.15, 10.43, 9.82. So approximately in the range of 9 to 10, right? So once we reach this particular pH value, that is 9 to 10, that is a basic pH, then this amino group will also lose its proton, right? And the amino acid will be existing as an anion. And whereas the R groups, considering the R groups now, so the acidic R groups, like for example the aspartic acid, glutamic acid, they also have less pKa values because they have these acidic groups which release the proton first. So their pKa values you can see in the range of 3.86, 4.25, the acidic amino acids, right? Whereas the R groups of the basic amino acids, they have these amino groups which will release protons later, right? So that is why their pH have higher pH value, higher pKa values. For example, arginine 12.48, lysine 10.53, but histidine has a lesser pKa of the R group because of the particular structure of histidine, if you remember. So they tend to lose proton at a lesser pH, which is approximately 6. Okay, the pKr of histidine is 6. So these are the amino acids, these are the few amino acids which also have a pKr value, or the pKa of the R group, which also comes into consideration when we are titrating these particular amino acids, right? So for example, if we titrate aspartic acid, okay, first tell me if there is any doubt in this particular slide. Okay. Okay, so I hope this particular point is clear, that when the amino acids are negatively charged from -1 to 0 to +1, how the charge of the amino acids are changing, and when the particular groups, the amino groups and the carboxylic groups, are losing their proton. Alright, next. So we are talking about this R, the pKa value of the R group. So that is why we will consider this titration of aspartic acid, which have 3 pKa values. Okay. Now, in the basic pH, the aspartic acid exists in this form. So what is the net charge on this molecule? Please tell me. Suppose when the pH is less than 2, what is the net charge on this particular molecule? See here, the carboxyl groups are protonated, so they do not have any charge, right? But the amino group is also protonated, which means it has a positive charge. So the net charge on this molecule is +1. Am I right? Okay. Alright. So now when we increase the pH, so the first pKa value, you will see that it is 2, right? So here this pKa is for the alpha carboxyl group, right? So pKa for the alpha carboxyl group is 2 approximately. So when we increase the pH to 2, then what happens? This alpha carboxyl group, it loses its proton, right? And now what is the net charge on this molecule? So we have NH3 plus and we have COO minus, both on this molecule. So now what is the net charge? 0, exactly, because this is a zwitterionic form. And now what happens, so again you can see the next P, so this is Pi, don't consider the Pi for now. Next pK2 is 3.9. Pi we will calculate, we will come to. But the next pKa value, which is pK2 for the carboxyl group of the side chain. So now the pKa of the side chain carboxyl group is 3.9. So when we increase the pH to 4, then what happens? this side chain carboxyl group also loses its proton, right? So here it was protonated at a pH which is um, 2, for example, right, or pH 3. But when it reaches the pH to 4, then what happens is the pK of the side chain carboxyl is 3.9. So at pH 4 or greater than 4, it loses its proton, the side chain carboxyl group. So now you have a side chain carboxyl which is CO minus, the alpha carboxyl also CO minus, and the NH3 group exists as NH3 plus. So now what is the net charge? -1. Very good. Now next what happens, it has another pKa, right? It has the third pKa value which is for the alpha amino group. The pKa for the alpha amino group is 9.8, right? So when the pH reaches 10, right, or greater than 10, then what happens, this alpha amino group also releases its proton. So now from NH3 plus it comes as NH2, right? So we have NH2 and we have 2 carboxyl groups existing as CO minus. So now again, what is the net charge on this molecule? -2, exactly. So here you can see that since it has 3 pKa values, this aspartic acid, so it exists in this +1, 0, -1 and -2 charge states, right? But how to calculate the pi of this aspartic acid? So remember, when we have amino acids which have greater than 2 pKa values, that means it has a pKa for its side chain as well, then what we do, we calculate the pi for this region of +1 to 0 to -1. So that is the charge of the molecule, right? So when the molecule is charged as +1 to when it is changing to 0, and finally when from 0 to it is changing to -1, this is the zone that we consider for calculating our pi. So that means the pKa values which are responsible for changing the charge of the molecule from +1 to 0 and from 0 to -1, those two pKa values we will consider for calculating the pi. That means the average of these two pKa values will be actually our pi, right? So here, can you tell me how to calculate this pi of aspartic acid, or what will be the pi of this aspartic acid? So you have to consider what? When the molecule is changing from +1 to 0, what is the pKa which is allowing this change of the charge from +1 to 0, and what is the pKa which is allowing the change of 0 to -1? So take a look at this structure and tell me that which are the pKa values. From +1 to 0 to -1. Yes, but I'm talking about what is the pKa value. So the first, here you see the first molecule, it has a +1 charge, right? Sorry, this has a +1 charge, and after pK1, after this pH pK1, pK1 is what? pK1 is 2, right? So due to this pK1, when the pH is increasing than that of pK1, then what happens? This molecule is losing its proton from the carboxyl group, and it is changed from +1 to 0, right? So what did I tell you? That you have to consider the pKa which is responsible for changing the charge of the molecule from +1 to 0. first. So the first pK which will come into consideration here is the pK which is responsible for changing the charge of the molecule from +1 to 0. So what is the pK here? That is pK1, which is 2. Tell me if I am right. Okay. Alright. So that is the pK1. Now what did I tell you next? Now the next pK that you have to consider is the pK value which is responsible for changing the charge of the molecule from 0 to -1. Right? So from 0 to -1. Right? So the charge of the molecule, when is it changing from 0 to -1? What is the pK? Please tell me. Which is making the... Yes, exactly. Very good. So this one is the pK2, 3.9, which is the pK2 value or the pH value where the charge of the molecule is changing from 0 to -1. Alright. So therefore, we will have to... So we have got this zone from +1 to 0 to -1. Right. So now we have to make an average of these two pK values, which will give us the pI of the aspartic acid. So therefore our pI will be half of pK1 plus pK2, which is half of 2 plus 3.9. So please calculate. Yes, approximately like that. So it's around 2.8 or something, right? So that is the pI of the aspartic acid, right? So here again I'm just going through it once more. To calculate the pI of aspartic acid, we have to consider, or any amino acids which have the 3 pK values, we have to consider the zone where the molecule is changing from charge +1 to 0 and from 0 to -1, right? And those two pKs we will average to calculate the pI of that amino acid. And here we have got the pK1, which is 2, and the pK2, which is 3.9, and we have got the pI of the aspartic acid as around 2.8. Right, so that is how we were calculating the pI of this aspartic acid. All right. So next we will look at another amino acid, which is a basic amino acid, and the lysine. Right, so we are looking at the titration curve of lysine. So this is how the lysine structure looks like. Is a big structure, right? And this is the R group, right? And here it is given that the pK1, that is the pK of the carboxylic alpha carboxyl group, is 2.22, 2.2, and the pK2, that is the pK value for alpha amino group, is 9.0, right? And next we have the pK for the R group, which is pKr, which is 10.5. All right, and this curve, the titration curve of lysine is also given. Now can you calculate the pI of lysine from this information? You have given the structure of lysine, and you are also given the pK values. So now what you have to consider is when the lysine is changing from +1 to 0 to -1 charge. Okay, and can you tell me how did you calculate it? Okay, so therefore here let's see how this +1 to 0 to -1 structure will change. Okay, all right, the charge will change. So the pk1 is 2.2, so obviously the molecule will lose its first proton from the carboxyl group, right, at pH greater than 2.2. So when this carboxyl group will um, release its proton, so it will exist in minus, okay, let's see here. I'm drawing a simpler structure of the of lysine. Okay, so this is the R group. Okay, this is the alpha amino group, this is the alpha carboxyl group. So when the pH is approximately greater than, or it is greater than 2 or 2.2, then what will happen, this alpha carboxyl group is going to lose its proton. So now the net charge on this molecule is +1 +1 -1, right? So which is the net charge? So +2 -1, which is +1, right? So the net charge on this particular molecule is +1. All right. Now next is, the next pKa value is pH 9, pK 9. So at pH greater than 9, the alpha amino group is going to lose its proton. So when we increase the pH, what happens? The alpha amino group is going to lose its proton. So it is going to be like this. So this is R group, right? So here you have CO minus NH3 plus, and this exists as uncharged NH2, right? So what is the net charge on this molecule again? It is 0. Very good. So now we have got this pH where it is changing from +1 to 0. Okay, and next what will happen? Next is this pKr, the R group will change. So the R group will lose its proton. So I just didn't tell you one thing, that is when we are beginning the titration, what is the in the most protonated form, what is the charge of the lysine molecule in the most protonated form? Sorry. Right, so at a lower pH, at a very less pH, this is the most protonated form of lysine. When the alpha amino group and the alpha carboxyl group, the alpha amino group and the side chain amino group, both of them are positively charged, right? And the alpha carboxyl group is having this proton, so it has no charge. So what is the charge that we are beginning with? It is +2, right? So the first pKa, which is changing the charge from +2 to +1, is 2.2, right? We know this pK1, 2.2. It is changing from +2 to +1. Now at pKa value, which is 9, that is pK2, this molecule is changing from its +1 to 0 when the alpha amino group is losing its proton. Okay, next is when the R group is losing its proton. So then what will happen? The molecule will exist like this. So R group is losing its proton, so it's losing its charge. The amino group has already lost its proton, so it is also uncharged, and the CO minus group has also lost its proton. So now what is the net charge of this molecule? Minus 1, right? And what is the pK here? 10.5, which is the pKr, right? So now let's look at it again carefully. When the molecule is changing from +1 to 0 to -1, and what are the pKa values responsible for that? So when is the molecule is in a +1 state? Here, right? That is the +1 charge state of the molecule, lysine. So the pKa, or the pK2, which is responsible for change in this +1 to 0. So that is 9, right? The pK2 value is 9, which is allowing it to change from +1 to 0. Or when the pH is increasing greater than 9, then this amino group is losing its proton, and now this molecule is having 0 charge. So the first pKa is 9. So now from 0 to -1. So now when the side chain amino group is losing its proton, then the pK or pH value is greater than 10.5, because the pKr is 10.5, right? And now this molecule is existing in a -1 charged form, right? So the pH or the pKa responsible for that is 10.5. Am I right? Okay, so now therefore what will be the Pi? Half of P. Yes, half of pK2 plus pKr, which is 9 plus 10.5 divided by 2, right? So approximately 19.5 divided by 2, which is approximately we can say 9.8, right? Okay. So Shuman, you understood what was the mistake you did first time? Yes, yes. So always you have to, you don't have to hurry here. Always you have to consider the structure, because this is a basic amino acid. The first state is this, the most protonated state is the +2 charge. So which is why this particular pK1 value you cannot consider while considering the calculation of Pi, right? So always you have to check the structure, and you have to check the charge when it is changing from +1 to 0 to -1. And so therefore by this we can determine the Pi of lysine, which is 9.8, which is the average of pK2 and pKr values. All right. Is there any doubt from anyone else? All right. Okay. So that's it. That's all for the discussion of today's session. Now we will move on to the question answers or the problems. Okay. So let me know your answers in the chat box. Please type your answers in the chat box. Or you can tell me also, like, by unmuting your mic. That's also fine. Okay, Shumon, I can tell you this answer is not correct. Please reconsider once. Check once more. Any answers from Aarti and Pushpendra? Non-polar and aliphatic side chains. Not C also, because C are the amino acids which are basic amino acids. So they are polar. They have charged. Their their side chains are charged. So the correct answer is alanine and leucine. Uh, should I go back to the structure once and tell you again? Alanine and leucine. Here. So this is how alanine looks like. So you can see that its side chain is aliphatic and it is also not polar. And leucine also same. So these two are aliphatic and non-polar amino acids. There is no polarity in the side chain. Okay. Let's move on to the next question. The correct solution is B here. Okay. Next question. So net negative charge at pH 7.4. So pH 7.4 is the physiological pH. So which are the amino acid I told you which have net negative charge at physiological pH, or negatively charged amino acids? Yes. Shuman and Aarti, answers? Okay, let's see. So the correct solution is A, which is glutamate and aspartate, not glutamine. So here there is a difference between A and B, that is glutamate and glutamine. So glutamate has this, what is the R group? I'm writing it here. Glutamate has this R group like this. This is the R group of the glutamate. And what is the R group of glutamine? I told you that glutamine is the amine of glutamate, right? So in place of COOH, it has CONH2. Okay, and because it does not have this dissociable hydrogen, right? So this proton is not present in glutamine, which is why glutamine is not negatively charged at physiological pH. So the correct answer is glutamate, and the other one is aspartate. Aspartate has this R group CH2 COOH. So just one methyl group is less than glutamate. Okay, so the two Negatively charged amino acids at physiological pH are glutamate and aspartate. Solution is A. Okay, moving on to the next question. You should be able to answer this. Um, can you tell me more clearly? I am not being able to hear properly. Isoelectric point, yes. Is that clear with the the rest of you? The pH at which the amino acids do not migrate in an electric field is called the isoelectric point. Because why? Because at the isoelectric point the net charge is 0. Yes, correct. Okay, next question. Which of the following is an amino acid? I discussed this during the aliphatic side chain amino acids. Okay, okay. So the correct answer, Shuman? Leucine? No, not leucine. The correct answer is proline. Okay, let's revisit the structure once again. It's okay, it's not easy to remember all the structures of the amino acids on the first go. So you need to get acquainted with the structures of the amino acids. Okay, so this is proline. And I told you this is an amino acid because this is an amino group and not an amino group. Okay, so you can see that this amino group is basically linked with the alpha carbon here, joined with the alpha carbon with a ring structure, so that's an amino group. So that is why proline is called an amino acid. Okay. Moving on to our next question. This is answer proline. Okay, question number 5. For this also you will need the help of the structures. Number of chiral centers in isoleucine and leucine. So you know what is a chiral center, right? Chiral center or chiral carbon. Okay. Answer, Pushpendra, sorry. Did you tell me the answer? Okay, I'm drawing for your help, for all of your help, I'm drawing the structure here. Okay. So that now you can figure out what are the number of chiral carbons. So first I'm drawing isoleucine. So this is isoleucine and this is leucine. Okay, and chiral carbons are the carbons which have 4 different groups in its 4 valencies, right? So it is associated with 4 different groups. So now you tell me what are the number of chiral carbons in isoleucine and leucine. Okay. Okay. Shumon is also saying B, right? All right. So let's see. What are the chiral carbons here? In isoleucine, this one is the first chiral carbon, and this is another chiral carbon, right? So isoleucine have two chiral carbons, and in leucine, the only chiral carbon is this one, the alpha carbon, right? So the number of chiral carbon is 1. So the correct answer is B, 2 and 1. Okay. All right. Moving on to the next question. This is a very easy calculation. Please calculate and tell the answer. You have to calculate the PI. Okay, I've got your answer, Shuman. Okay, D. Right. So the PI is half of 2.34 plus 9.60, which is approximately 5.97. So D is the correct answer. Yes. Okay. Next question. This is also a structural information. Why are tyrosine and tryptophan less hydrophobic than phenylalanine? Okay, I've got the answer from Shuman. Any other answer? Yes, Pushpendra. What is your answer? C. No. The correct answer here is B. Okay, let's go back to the structure once again. So you see that you have to remember that all the structures of the amino acids to answer these kind of questions if you are attending the exam for this course, or for other like studies also, your studies also, you need to remember the structures of these amino acids. Okay, the aromatic amino acids. This is phenylalanine, this is tyrosine, and this is tryptophan. So the question was, phenylalanine is more hydrophobic, right, than tyrosine and tryptophan. Why? So the answer remains in this structure. You can see that in tyrosine and tryptophan there are polar side chains. So because the tyrosine has this OH group, so it gives some polarity to this structure, right? So because of this phenol group present. And here in tryptophan also, since it has this indole ring, so this nitrogen is imparting some polarity to the structure, right? Which is why their hydrophobicity is less than phenylalanine, because you can see that there are no polar groups present in the side chain of the phenylalanine, only a benzene ring and a methyl group, which both of them which are not polar, right, which are not hydrophilic. So phenylalanine is more hydrophobic than these two amino acids, tyrosine and tryptophan. Is that clear, Pushpendra, also? Okay, oh, all right. Moving on to our next question. So, any idea about the pi of alanine? Pi of alanine is approximately 6. I'm giving you one hint here. Pi of alanine is approximately 6. So, what will be the charge on alanine when the solution pH is 7.3? Negative. No, actually not. So I know from which point of view you are telling that it is negative, because when um, pH is greater than pi, then the amino acid is negatively charged, and when pH is less than pi, then the amino acid is positively charged. That is true, but here the if you look at the titration curve of alanine, you will see that in glycine also you saw one point where there was a sharp rise like this, right? This kind of a form. So what was that? So that is basically when pH is increasing, but the net charge on the glycine molecule is not changing, right? So that is the buffering zone of this amino acid. So this pH 7.3 is also similar in that case, because the alanine reaches its zwitterionic form in pH 6 and it maintains the zwitterionic form in pH 7.3 as well. So the correct answer will be zwitterion. Okay, but if the charge was if the pH was approximately like 9 or something, then this alanine will lose its uh, the alpha amino group proton, and then its charge will change. Am I correct? The when will the charge of the alanine change? When only when the alpha amino group will lose its proton, right? So then only the charge from 0 to -1 will occur. So the change will occur. So therefore at pH 7.3 this change does not occur, which is why alanine at pH 7.3 will remain as a zwitterion only. Is that clear? So the correct answer is a zwitterion. Okay. Next question. So we have studied the titration curve of lysine, and what are the number of inflection points in this titration curve? Inflection points means the number of the sharp changes in pH. How many inflection points do you observe? Yes, Shuman, your answer is correct. We observe two inflection points. So the curve looks something like this. That isn't it? So there are these two inflection points. So the correct answer is 2. Okay. Okay. Next question. Which of these amino acids have pI close to physiological pH? Think about the properties of these amino acids which are given in the option. Like first one is asparagine, next is serine, the third one is histidine, and the fourth one is glutamine. What are these? Yes, please tell me again. C. Okay. Any other answers? Suman, are you saying D? Glutamine. The correct answer here is histidine. So histidine pI is approximately 7.6. So of course at a physiological pH which is pH 7.4, the histidine will exist. So this has a very the pH, the pI is close to physiological pH, right? And asparagine, glutamine, these are amine containing amino acids, right? So their uh, pI is not close to the physiological pH. It does not come in the range of physiological pH. Okay. Moving on to our next question. Okay, here there is some calculation involved. Let's do it. Arginine. Okay. The amino acid is arginine, so which is a positively charged amino acid. So it has amine group in its side chain as well, right? So now tell me what will be the pI of arginine. Think about the structure. You don't have to know the full structure, but from the general overall structure also you can get this idea. And you know how to calculate the pI. +1 to 0 to -1, right? Okay. I've got your answer. B. Okay, right. So that is the correct answer. And why is that correct? Because the in the most protonated form. Yes, Pushpendra, sorry. Please tell me again. You are not actually very clearly audible. Can you raise your volume and tell me? Little bit. You are not audible, very sorry. Um, so as I was saying, that when so the pKa of the alpha carboxyl group is 1.8, alpha amino group is 9, and side chain pKa is 12.5. So in its most protonated state, arginine will exist as COH, NH3 plus, and in the side chain also NH3 plus, like this. So in the most protonated state it is plus 2. When it is changing from plus 2 to so what will happen first is that the as we increase the pH, the first proton will be lost from COH, so it will be converted to CO minus, NH3 plus, NH3 plus, just like lysine we were talking about. So when the net charge will be plus 1. Okay, so now we've got the net charge plus 1. So this pK was 1.8. Next is at pH greater than 9, the alpha amino group will lose its proton, so it will be NH2 CO minus NH3 plus. So the net charge will be 0 on this molecule. So the pKa responsible for that is 9. Now next is when the at pH greater than 12.5, the arginine molecule will lose its proton from the side chain as well. So now the net charge will be minus 1. So from plus 1 to 0, pK is 9.0. 0 to minus 1, pK is 12.5. So we need to make an average of that, which is 10.75. So that was your answer, which is the correct answer. Okay, 10.75 is the PI of arginine. Very good. Okay, moving on to our next question. Shruman D, are you sure? What is the role of HCl? We were discussing during the titration of glycine. So when we dissolve glycine in water, the glycine exists in zwitterionic form, because glycine in physiological pH it exists in a neutral pH, it exists in a zwitterionic form. So when we need to titrate glycine, that is we have to protonate the glycine, we have to lower the pH. Okay, so that is why we are adding HCl. So the correct answer is Arthi is one, that is correct. It completely protonates glycine. Okay, so the role of HCl here is that when we are beginning the titration, we have to reach a completely protonated state of glycine. Okay, because otherwise we cannot measure the change in pK, right, or the pH or the pKa. We cannot, initial pK we cannot measure. So to completely protonate glycine we need HCl, because HCl is acidic, it will lower the pH, and once it lowers the pH, the carboxyl group, alpha carboxyl group, will lose its proton. And when the alpha carboxyl group loses its proton, then what happens? Then it is not the alpha carboxyl group loses its proton, but at pH lower than that, what happens? The amino group is positively charged, and the alpha carboxyl group is does have the proton, so not loses its proton, but it does have the proton, right? The alpha carboxyl group has the COH, and the uh, alpha amino group is also NH3 plus, right? So at low pH, right? This is at low pH, but at physiological pH or at pH 7, pH 7, what happens at the pH of water? Then glycine exists in this kind of form, right? And this is a zwitterionic form, so this has zero charge, so we cannot calculate or we cannot begin titration at this point. So we need a lesser pH, because of which we add HCl, which protonates glycine. Okay, so this hydrogen atom is added, this proton is added to glycine. That's the role of HCl. All right, moving on to our next question. But this question is a little bit different. Think about it. So suppose the pKa for the side chain of histidine is 6 when it is in isolated state. So when it is not inside any protein. But suppose there is a protein and there's an active site of the protein, right? The catalytic site or the active site of the protein where histidine is present, and suppose there are other amino acids which are present in the active site which are also charged. They can be positively charged, they can be negatively charged. So how the presence of these positively or negatively charged amino acid is going to affect the pK2 of histidine, because they are very close together. So what happens in an active site, so the protein exists in a three-dimensional conformation, right? So in an active site, many amino acid residues are very close to each other. Okay, and if the amino acids are close to each other, then there will be either interaction between them, like positive charge, negative charge, electrostatic interaction or electrostatic repulsion. There can be many things that can be occurring in the active site region, right? Or any region of the protein where the amino acids are close together, right? So for example, in the active site, if there are positively charged amino acid, suppose positively charged amino acid around histidine. So histidine, what is the charge on histidine? Histidine is also like a basic amino acid. Okay, so the, it is also positively charged, for example. Now what happens that if there are positively charged amino acid residues close to it, then what happens that these positive charges destabilize the positive charge on histidine. Okay, and so which is why it becomes difficult to deprotonate the histidine or dissociate the proton from the histidine side chain. In this condition, when there are many positively charged amino acid residues close together, it destabilizes the positive charge on the side chain of the histidine, which is why it is difficult to deprotonate the side chain of the histidine, which is why it requires more amount of like or what to say, it requires less pH. Okay, so if it requires less pH to protonate the histidine residue, that means, so protonate means what? To add the proton, right? So to add the proton in the side chain of the histidine, when there are many positive charges around the histidine, so then what happens, it is difficult to add one more positive charge in that, right? Because it will increase repulsion. So to protonate histidine at this point, when there are positive charges around histidine, so it requires less pH. So if it requires less pH, it means the pKa will also be less, because you know pKa will also fall, um, which is why the pKa of the histidine will be less than 6 if the active site has more positive charges. So that is the correct answer here. So did you understand this? So if there are positive charges surrounding histidine, then it is difficult to protonate histidine. And if it is difficult, that means it requires less pH. So more protons should be present in the solution to protonate the histidine side chain at this position. So more protons present means less pH, and less pH means less pK. If less pK, that means the pK2, that means the pK of the side chain will decrease in this situation. So it will be less than 6 if the active site has more positive charges. Okay, so that is the role of the surrounding amino acids, which is why in a protein it's not very easy to determine the pKa of the side chains, or the pKa of the alpha carboxyl group or amino group. The alpha carboxyl and the amino groups, they form the peptide bond, but the side chains are free. But the determination of pKa of the side chain is difficult, because in a protein it exists in a complex form. So a protein exists in a three-dimensional form, right? So there are many charges surrounding, there are many amino acids surrounding, so there are many interactions, right? So which is why it is not easy to determine the pKa in this kind of a condition. Okay, moving on to our next question. So there is a situation where there is the change of pH is very slow. So you are keeping on adding alkali, but the change of pH is very slow. Why does that happen? So for example, the curve, if you remember, so here you are adding alkali, right? OH minus ions. Here this is the change of, this is the pH axis. And what happens that, okay, so it becomes like this kind of a position, this kind of a zone. What is it called? No, D is not the correct answer. So here what you are seeing, that you are adding alkali, but the change in pH is slow. So this is actually the buffer zone. This is actually the buffer zone of glycine. That is the correct answer here. Sorry, I'm just mistaken a little bit here. So here actually the buffer zone, I'm just confusing it. So this is actually the buffer zone, this part, where you are keeping on adding alkali but change in pH is very less. Not this one, I'm very sorry. This one is where the change in pH is occurring very fast. But this particular zone, this is the buffer zone. When you are keeping on adding alkali, but you can see that in the y-axis there is less change. So this is the buffer zone in the graph of titration. Alright. Okay. And the last question for today. So this one will have multiple answers. So arginine, when it is present in a protein, so first of all, there are many other amino acids which make the process of determination of pi complex. And also when you have a protein, so what happens is that the um, the structure of the protein might get damaged or affected at a higher pH. So the correct answers are B and C, that there are multiple amino acids together, so the determination of the pi is difficult or titration is difficult, as well as when you are increasing a pH, the protein will lose its structure. So the titration is difficult, or it's not easy like the individual arginine residue, right? Okay, so do you have any other doubts from today's class? All right, so that's all for today. Thank you all for joining. And this is the YouTube channel where you will get the recordings of all these live sessions. And thank you all. Next Tuesday, we will again meet 6 to 8 p.m. for the week 3 live session. Thank you all for joining.